Hi everyone, welcome to topic 4, the genetic blueprint and energy production. So in this topic, we're basically going to learn about how, everything requires energy. So you are learning about energy production right now, and that takes energy to learn. And so we're going to learn about that process, how we use energy while thinking, how our body uses energy to do everything. Even sleeping, we require energy. And so we're going to talk about how for humans, we get our energy from the foods that we eat. So by taking in nutrients from our foods and breaking them down and metabolizing them, we can actually convert that chemical energy into something called cellular energy, or ATP. So we'll learn about that process in this topic. We will also learn about how that process is dictated by our genetic code, or DNA. So basically the DNA is what basically codes the proteins and tells the body what to do. So we'll go ahead and talk about that as well. So to start us off with topic 4, we're going to start out by looking at how DNA is structured. So by the end of this video, you should be able to list the 3 major subunits that make up nucleotides, list the 4 nitrogenous bases found in DNA, explain the primary structure and function of DNA and ATP in a cell, as well as describe how eukaryotic and prokaryotic DNA is arranged in the cell, as well as identify the complementary base pairs of DNA. And just like all the other topics, I highly recommend that you write down these learning objectives and answer them at the end of the video as a review, okay? And then use that to help you kind of prepare for the exams. So before we get into how DNA is structured, it's important to learn a little bit about the discovery of DNA. So a lot of it is credited to Watson and Crick. So in the 1950s, Watson, Francis Crick and James Watson, they determined its structure, so that double helix shape of the DNA structure. But it was a group effort. It wasn't just them that discovered its structure. There was a lot of people that are involved with putting the pieces together. Okay, so basically they were just the ones that kind of brought it all together in the end, but there were a lot of pieces involved. So there were other people that were involved, such as Pauling and Wilkins. They were the ones that discovered the proteins involved. So they used a process known as crystallography. There was a woman involved, a woman researcher who worked in Wilkinson's lab. Her name was Rosalind Franklin. And her data was actually very important in helping Watson and Crick in determining the structure. And so, her piece of the puzzle actually helped them kind of solidify their idea of this double helix structure of the DNA molecule. In 1962, Watson, Crick, and Wilkins were awarded the Nobel Prize in Medicine. Unfortunately, Rosalind Franklin was not awarded because she had died in 1958. And this was prior to the Nobel Prizes rules, where I think it was like in the 1970s, where you could receive a Nobel Prize after death. But since this was prior to that rule, she did not receive the Nobel Prize because obviously she was dead at the time. But she well deserved it. Okay, so let's get into how it's structured. So let's begin with a little bit of review of the monomer. So we recall in the last topic, topic 3, we learned about the 4 macromolecules of life. So we learned about the carbohydrates, the lipids, the proteins, and the nucleic acids. So we spent a little bit of time talking about nucleic acids, the DNA and the RNA, and those being the polymers and the nucleotide being the monomer that makes up the polymer. So the nucleotide is the subunit or monomer of the nucleic acids. So if we're talking about just strictly DNA, the nucleotide of DNA is made up of 3 parts. We have a sugar, a phosphate group, and a nitrogenous base. And if we want to get a little ahead of ourselves, RNA also is made of a sugar, phosphate, and nitrogenous base. Okay, so let's take a closer look at that word DNA. So DNA stands for deoxyribonucleic acid. So we know it's a nucleic acid. But if we look at the first part of that word, deoxyribo, well, that means a sugar, okay? So the nucleotide is, made up of 3 parts, a phosphate group, a sugar, and a base. So that sugar is a deoxyribose. So deoxyribose is actually a 5-carbon sugar. So if we remember back in topic 3, we talked about carbohydrates. So carbohydrates are sugars, right? And we talked about how the monosaccharides, they can range from having 3 carbons all the way up to 6 carbons. So in our DNA, the sugar that our DNA has is a 5-carbon sugar, which we call deoxyribose. Okay, and so that is part of the name deoxyribonucleic acid because it contains the sugar deoxyribose. And again, if you want to get ahead of yourself, you can think of RNA. RNA is ribonucleic acid because it contains a different 5-carbon sugar called ribose. Okay. Also involved with our 3 parts of our nucleotide, we have the phosphate. Okay, so that's the phosphate group. And then on the flanking the other side of the sugar is the nitrogenous base. And for DNA, we have 4 possible nitrogenous bases attached to the sugar. It could be an adenine, which you can see down here, a guanine, a little bit different, a cytosine, or a thymine. Okay. Now, if you take a look at the 4 different nitrogenous bases, we can categorize them into 2 kind of subgroups based on how many rings they contain. Okay. So if they're a purine, they contain 2 rings. So purines, 2 rings. That kind of rhymes, right? Purine, 2 rings. So guanine and adenine have 2 rings. So purines have 2 rings. And then the pyrimidines have 1 ring. Okay, so cytosine and thymine have 1 ring. And we'll get into a little bit more about the pyrimidine and the purines when we talk about complementary base pairing. Because later on, we'll learn that a purine must pair with a pyrimidine. Particularly a guanine pairs with a cytosine, an adenine pairs with a thymine, just because it kind of evens it out. All right. So that's, the basic structure of the nucleotide or monomer of our DNA polymer, okay, the nucleic acid. Now again, we talked about how Watson and Crick, they figured out that DNA is a double helical structure. So it consists of 2 strands of nucleotides. Okay, so if we take a look at this picture, we have 1 strand of DNA, and then we also have another strand of DNA. So you can see how the 2 strands, they basically kind of twist in a pattern that makes it look like a helix. Okay, so we kind of call it a helical structure. So it's a double helix. And if we look at the strands, the way that they're arranged is that if I untwisted it, it kind of looks like a ladder. Okay, so the sides of the ladder, this part, which is, you know, pretty strong, this consists of the sugar and the phosphate part of the nucleotides. Okay, so if I go back a slide, so we're talking about the sides of the ladder. Okay, so the sides of the ladder are held up by the sugar phosphate part of the nucleotides. So if I have a sugar and a phosphate of one nucleotide, that phosphate group of one nucleotide will bond with a sugar of a nearby nucleotide. And that sugar will bond with another phosphate. So it forms this kind of sugar phosphate backbone. So when we're looking at this double helix, the sides of the ladder are made of the sugar phosphate backbone. So it's that forms the backbone. The third part of our nucleotide, the nitrogenous base, forms the steps of the ladder. Okay, and this is what holds the two DNA strands together. Is the nitrogenous base of one DNA strand and basically a bond that forms between the nitrogenous base of the other DNA strand. Okay, so one nitrogenous base of one strand forms a hydrogen bond with the nitrogenous base of the other DNA strand. And they form what's called a complementary bond or base pair. Okay, and they form this weak bond, which is a hydrogen bond. So remember, hydrogen bonds, they're weak bonds. And so you can see that they're not always going to be fully attached. Okay, so they're weak bonds, so they will be, you know, attaching, detaching a lot. But the sugar phosphate backbone, the sugar from one nearby nucleotide attached to the phosphate of the other nucleotide, are held together by covalent bonds. And because they are held together by covalent bonds, those are strong bonds. And so the backbone is strong. The rungs of the ladder are held together by hydrogen bonds, which are weak. Okay. Now, when we're looking at the base pairs, the nitrogenous base pairs of one side of the ladder will complementary base pair will pair up with the other side. And the way that it's arranged is if we have an adenine on one side, it will always pair up with a thymine on the other side. Okay, so A always pairs up with T. A will never pair up with G, it will never pair up with C, it always pairs up with T. So the A's and the T's always pair up. Okay? Guanine always pairs up with cytosine. So you'll never see guanine pair up with thymine, you'll never see it pair up with adenine. It always pairs up with cytosine. Okay? So the A pairs up with the T, the G pairs up with the C. You'll never see them pairing up with anything else in a DNA double helix. Okay? So that is our double helix structure. So it's composed of two DNA molecules held together by these complementary base pairs that have hydrogen bonds linking them together. So let's take a closer look. So imagine here again is our double helix. So we have our sugar phosphate backbone. So here is the sugar phosphate backbone. We can see here is one sugar phosphate backbone. We have the sugar part and the phosphate part of one nucleotide. So you can see the phosphate part is attached to the sugar part of a nearby nucleotide. Here's a sugar part, there's the phosphate part, it'll attach to another nucleotide. Okay, so this is forming the sugar phosphate backbone. And what holds the phosphate to the nearby sugar is a covalent bond. So this is a strong bond. Okay, it's a special type of bond called a phosphodiester covalent bond. It's just a covalent bond. It's got a special name, but that's what holds the sugar phosphate backbone together, the two nucleotides together. Okay, and you can see it also on the other side. So here's the other side. You have the nucleotide, you have the sugar and the phosphate of one nucleotide. The phosphate group is attached to the sugar of the other one. Okay. You can see the nitrogenous bases. Here is a thymine of this nucleotide, and it's going to form a hydrogen bond with an adenine of the other DNA strand. Okay, so these are weak bonds that are holding them together. Here's a guanine. Guanine is going to remember it only bonds with cytosine. So it's going to form hydrogen bond with a cytosine on the opposing strand. Okay, so now let's get into a little bit more detail about the actual structure, how it's arranged, because it's arranged in a very interesting way that will help us in the future when we're talking about like DNA replication and so forth. So I'm going to get my pen out. And when we look at this pentose sugar, remember it's a 5-carbon pentose sugar. It's a pentose shape, right? So we start with this oxygen in the center, and then at each corner is a carbon. So this is carbon number 1, this is carbon number 2, this is carbon number 3, this is carbon number 4, and then up here is carbon number 5. Down here, here's the other nucleotides. So here's the first carbon, here's carbon number 2, here's carbon number 3, here's carbon number 4, and here's carbon number 5. Right? Number 3 looks kind of weird there. If we look at the opposing strand, it's kind of in the opposite direction. So here's carbon number 1, carbon number 2, carbon number 3, carbon number 4, and carbon number 5. And then same thing, 1, 2, 3, 4, and 5. Now the reason why I'm putting these carbon numbers in is because when we're looking at this strand, we can see that it's running in opposite directions as this strand. Okay, so we call this end the 3' end, because this is where the third carbon is facing. Okay, and we call this end the 5' end, because this is where the 5-carbon end is facing. Okay, and it's running the opposite as this side. So notice the 5' end is facing this direction, the 3' end is facing this direction. So we call that anti-parallel. So the two strands are running anti-parallel to each other. Okay, so that is a little different, right? Okay, so this is something that will come into play also when we're talking about DNA replication. Because if you notice that 3' end, look at what's attached to the 3 carbon, an OH group. So this is where the next nucleotide is going to attach, because there's going to be a what type of reaction occurs when we remove water to bring together two monomers? Dehydration synthesis. So we're going to remove water. So we're going to actually add a nucleotide, and it always adds on the 3' end. Okay, we can't add it up here. So we always add it at the 3' end. So that's another clue. So we always add it at that 3' end. So we'll talk more about that later. But just, this kind of proves that point that it's running in opposite directions. So that's why you'll see the 3' end. That just means that third carbon, okay, is, there. And then if we follow that up, that means that the 5th carbon is on the top, okay? And then the other strand is going in the opposite direction. So it's going anti-parallel to each other, okay? So now we're just going to go into a little bit information about the RNA structure. So the RNA structure is slightly different than the DNA structure in a couple ways. So the first way is that the RNA stands for ribonucleic acid because it contains a ribose sugar instead of a deoxyribose sugar. So what's the difference? Well, deoxy, that sounds like deoxy. D means like remove, so remove an oxygen. So deoxy means one less oxygen. So let's take a look at our deoxyribose over here. So this is DNA sugar, this is RNA sugar. What do you notice that's different? RNA sugar has one extra oxygen. Okay, so there's one less oxygen in DNA. Okay, so deoxyribose, we removed an oxygen. Ribose, we still have the oxygen. That's the only difference, okay? The rest of the sugar is the same. And look at those carbons, by the way, they're labeled. So here's the first one, here's the second, here's the third, fourth, fifth. Okay, so that's one difference between DNA and RNA, is that we have one extra oxygen on the sugar, which makes it ribose. Phosphate group is the same, and it also has 4 nitrogenous bases, just like DNA in its nucleotide. However, the 4 nitrogenous bases, 3 of them are the same as DNA. They're still adenine, they're still guanine, they're still cytosine, but there's no thymine. So in replacement of thymine is a slightly different structure, which we call uracil. So uracil is a different nitrogenous base. Okay, it's still a pyrimidine like thymine, just a single ring, but it is different. It's differently structured than thymine. Okay, so we call it uracil. And then just like with DNA, adenine on RNA will bind with thymine on DNA. Guanine will bind with cytosine on DNA. Cytosine will bind with guanine on DNA. But RNA uracil will bind with adenine, okay? Because, adenine on DNA, it can't bind with thymine on RNA because there's no RNA, so instead it binds with uracil. Okay. And then the final thing that's different between DNA and RNA is that RNA is single-stranded, so it doesn't form a double helix like our DNA. Okay, so it's never double-stranded like DNA. It's always just one single strand. Okay. All right, so let's go ahead and do our review question. So which of the following does cytosine pair with? Is it adenine, guanine, thymine, or a pyrimidine? If you chose guanine, I would agree with you. So guanine is a purine. So cytosine is actually a pyrimidine, so it's not going to bind with that. So C binds with G all the time. All right, so that's it for our first part of topic 4, how is DNA structured? So by now you should be able to list the 3 major subunits that make up both the DNA and RNA nucleotides. You should be able to list the 4 nitrogenous bases found in DNA, as well as RNA. You should be able to explain the primary structure and function of DNA and ATP in the cell. Well, we didn't talk about ATP, but the primary structure and function of DNA in the cell contains the genetic information. ATP is involved with energy. Describe how eukaryotic and prokaryotic DNA is arranged in the cell. Hey, we didn't talk about that. But yeah, I guess we didn't really talk about it, and it's not in your textbook, but I guess we can talk about it right now. So if we kind of review way back into, gosh, was it topic 2, where is DNA found in the eukaryotic cell? It's found in the nucleus, right? Prokaryotes, like bacteria and archaeans, do they have a nucleus? Nope. So instead, their DNA is found in the nucleoid region of the cell. And with eukaryotes, our DNA is found on chromosomes, so those are kind of linear structured, whereas prokaryotic DNA is kind of circular. But we'll get more into that later. I think it's way over in topic, a little bit further along. And then you should be able to identify the complementary base pairs of our DNA. Okay, so for our next video, you will learn about how DNA is replicated, which is why we need to know that 3' and 5' ends. Okay, so we'll see you there.